This presentation is about a research paper on the microRNA expression profiling of male breast cancer, and it's presented by Mary Helder, Marvelous Matara, and Anastasia Philly. I'm just going to talk about the introduction of the research paper, of this research paper. First of all, I will talk a little bit, I will explain a little bit about the miRNA, and then I'll go on to the breast cancer and the methods. mRNAs are actually approximately 22 nucleotide small non-coding RNAs that modulate gene expression by binding to target mRNA by imperfect complementary, causing either mRNA degradation or translation inhibition, as shown in the Fig 1. Breast cancer is a rare disease in men, representing less than 1% of all malignancies and responsible for 0.1% of male cancer deaths. It has actually been indicated that the male's breast cancer, they are actually significant. They have shown significant morbidity and mortality. Meanwhile, microRNA gene expression analysis was used in this research as the experiment during the experiment, and it has been largely used to characterize human cancer. Previous studies have demonstrated that there is a large number of deregulated miRNAs in human breast cancer, and also about 3 recent studies have also proved that most of the deregulated miRNAs are involved in breast cancer metastasis. mRNAs can be potentially used as innovative and targeted therapeutics. Now I'm just going to pass it on to one of my colleagues who will tell you more about the materials and methods used in this in carrying the experiment in this research paper. To conduct the research, they had 23 cases of males with breast cancer and 5 cases of gynecomastia, and 10 female patients affected by ductal breast carcinoma. All these patients had undergone treatment by either simple mastectomy or wide local excision and radiotherapy. Right, the tissue samples taken from these patients were deparaffinalized with saline at 50 degrees centigrade for 3 minutes, and then RNA was extracted from all tissue samples. The tissue samples, the RNA extracted was hybridized on microarray chips, and 5 micrograms of those RNA of each sample were reverse transcribed using biotin and labeled random oligonucleotide primer. The biotin labeled complementary DNA was later hybridized again, and using microRNA chip, which contains 1,100 microRNA probes, including 326 human and 249 mouse microRNA genes in duplicate. So after carrying out the microRNA microarray, the images of the genes were analyzed, and statistical and bioinformatic analysis was carried out. In this method, mature miRNAs differently expressed between breast cancer and gynecomastia. They were identified using random variance t-tests, and genes considered statistically significant if their p-values were less than 0.01. This a quantitative real-time PCR was carried out, and where the mature mRNAs were dictated and the gene expression levels were quantified. Histochemistry analysis was carried out. In this method, the tissues were stained for vascular endothelial growth factor, that's the VEGF, and the homeobox 10, that's the HOD 10, and then lightly counterstained with hematoxylin. After this, the immunohistochemical score intensities were then recorded. In the few slides, I'll be discussing the results of this research paper. First of all, for the deregulated mRNA in male breast cancer to be identified, the mRNA expression profiles were determined for 5 male gynecomastia samples, 23 male breast cancer samples, and 10 female breast cancer samples using a custom microarray platform. All of the 23 male breast cancer samples pathologically corresponded in invasive ductal breast carcinoma. The link below, which is our table 1, therefore summarizes the clinopathological characteristics. This link below shows the second result that was derived from this paper, and it represents the different microRNA expression between male breast cancers and the cases of male gynecomastia. Opening this table, the p-value shows us the statistical difference in between the expression of mRNA in cancer and the gynecomastia, which was used as a control. The fold changes shows how much difference between the two stages of cancer. This link leads us to table 3, which shows the difference between the microRNA expression between male breast cancers and cases of male gynecomastia after the enrichment of the tumors. 13 genes were dysregulated. 3 of the mRNA genes were upregulated, which means they're positive, while the rest were downregulated, which means they're negative. P-values are also significant because they're all less than 0.01. More fold changes were also found between genes in gynecomastia and cancer. In summary, this study showed that there is a clear difference in the pattern in which microRNA are expressed in sufferers of gynecomastia compared to the ones with male breast cancer. This evidence can be used to differentiate between gynecomastia and breast cancer. It can also be used to differentiate between male breast cancer and female breast cancer. To improve this status, a large series of male versus female breast tumors can actually be used in order to address the possible role of microRNAs concerning the biological and prognostic tumor characteristics. This present report provides the first study on mRNA expression in male breast cancer, and the result derived from this research paper actually suggests that specific mRNAs may be directly involved in male breast cancer development and may be used as a diagnostic tool in the characterization of specific cancer gene targets.